Well, hello, welcome back. We're now ready to complete the examination, the review of cells. We've looked at the outer membrane, the nucleus, endoplasmic reticulum and Golgi apparatus, elements of the cytoplasm, elements like the cytoskeleton, outer cell membrane. We're taking a trip through the organelles that we find in the cytosol. And here's a dandy. This is the mitochondrion. It's odd in several ways. It's very important because this organelle in our cells, in the cells of all organisms that can use oxygen, this organelle is the site of aerobic respiration. That means this is where organic molecules that are nutrients are broken down to CO2 and water to produce ATP. All living things use ATP for their energy. What we see immediately when we examine the mitochondria is this outer enclosing membrane and an inner membrane that is very convoluted, folded back and forth, creating multiple spaces inside. So you can see there's the space between the outer membrane and the outer part of the inner membrane. There's also the space in the inner membrane. And that creates different reaction locations. So this is kind of a way of setting up a site for a special function. The conversion of nutrients to CO2 and water and the release of energy so we can make ATP involves many, many enzymatic steps. And that's the reason for the folding, greater surface area. So if we could blow this up enough, what we would see are enzyme systems embedded in this inner membrane surface. And what do I mean by that? Well, let's say you have a set of reactants and you have a product and you want a lot of that product. But there are 10 steps to get from the reactant to the product. Every step is catalyzed by its own enzyme. So if you just mixed that all together, here's what happens. The reactants are added, and after a while of diffusing around, they run into enzyme 1, and they go to intermediate 2. Now they have to float around till they find enzyme 2 and become intermediate 3. And that has to continue until you reach product. Not very efficient. However, if you take those enzymes, like the pointer is showing, and line them up 1 through 10, when the reactants find number 1, it goes through all 10 reactions straight to product. So it's like an assembly line. It's an efficiency improving device. And frankly, the more enzyme membrane surface area you have, the more enzyme systems you can have. So instead of having 10 enzyme systems, you can have 10,000. And as a result, the amount of ATP produced, the amount of organic molecules like glucose or fatty acids that are processed goes way up. Now here's another one of those amazing electron micrographs, 46,000 times magnification. Now we can see an organelle. By the way, what else can we see? See all these little dots? Those are free ribosomes. Do you see these folds here with dots along their edges? See it right there very well. That's a fold of endoplasmic reticulum covered with ribosomes. That's rough ER. But this color-enhanced photo, and by the way, this is artificial color, shows the clear structure that we've explained for the mitochondria. There's that double membrane, the outer membrane showing here, and then the inner membrane folded back and forth, producing those increased surface area structures. They call those cristae, and the inner contents, the gel of this inner part of the mitochondria, is called the matrix, and they contain all the chemicals necessary for aerobic respiration. But that's not the only interesting thing. about the mitochondrion. This was not originally a part of this surrounding cell. We see in the world around us many microorganisms living independently as single cells in the pond water, in the soil, in the air. And these show different characteristics. Some of these single cells can do aerobic respiration. They're what we call aerobic bacteria or aerobic microbes. If they can use oxygen, they can make lots of ATP out of organic molecules as well. Other microorganisms cannot do that trick. They're called anaerobic, and they make much less ATP, so they grow slower. At some point in history, those two types of cells combined, and an aerobic prokaryote took up residence inside an anaerobic one. Now, what are the advantages? The advantage to the aerobic is that the environment inside the cell takes advantage of the protection of the outer membrane, the consistent chemical environment of a living cell. That's a lot less variation to put up with. So it can grow and reproduce faster as it adapts to those conditions. It's protected. Whereas, what's the advantage to the outer cell? Well, the outer cell has now something living in it that's making a lot of ATP. If some of that ATP leaks out, or if this inner cell actively transports some of that ATP out, then it benefits this outer cell with an increased supply of ATP. So in time, this became an obligate relationship. The origin of this mitochondria was from the combination of two kinds of free living cells. It's called endosymbiosis, which means within, and cooperation. And in fact, this is not just the model and the accepted hypothesis for the origin of mitochondria as occur in our cells, but also the origin of mitochondria that occur in all eukaryotes that use oxygen. Also, a different kind of organism that was photosynthetic gave rise to the chloroplasts that now live inside plant cells. So this is a very robust and interesting feature. The cell itself cannot make a mitochondria. If you remove all the mitochondria from that cell, it cannot make another one. The other issue is that mitochondria actually carry within their insides some of their own unique DNA. Only mitochondria and chloroplasts perform this trick. This overall cell, most of its genes are in the nucleus, but a few are out here in the mitochondria, a remnant of those free living days. Now, when we start to look at genetic material, the organization of the DNA within the nucleus, we find an active cell stage followed by an inactive stage of DNA expression. When we look at a cell sample, most of the cells are non-dividing, and they look sort of like this, with a nucleus that is clearly different from its surrounding cytosol, but basically not much is visible. If we blow it way up, what we see are extended strands and tangles of what's called chromatin, staining fibers. And those fibers are made of the DNA double helix that are expanded out. Now, this DNA double helix, as we explained last time, as we'll talk about in a minute, this DNA double helix contains the genes and complementary base pairing and nucleotide base sequence codes for the production of a specific protein. It is this non-dividing cell, the intercell stage, when these DNA double helix are unwrapped, looking like this in the nucleus, that we are expressing genes and making proteins. When the cell has grown and matured, and basically stored up enough resources to divide, a change takes place where this DNA is wrapped up, it's bound up. It's sort of like if you went in the basement and found a bucket full of rope. The reasonable thing to do would be to stretch that out, untangle it, and then to coil it, or to produce, one of those binding wraps that holds the rope in a compact bundle so it doesn't get all tangled up. And that's what we're doing when we prepare this chromatin for cell division. We wrap it around proteins called histones, basic proteins, and then we coil those histone bundles. You see the supercoil here. Now, as you continue to wrap and bundle, just like coiling a rope, there are enzymes that take these coils and basically pack them in to a compact stage of the organelle called a chromosome. Now, when you take these really fine strands down here, and by the way, the scale between here and here is not really represented artistically very well. This is really lots and lots of DNA packed into this chromosome. You often see a kind of a pinch point. That's where the centromere is. The centromere is where we will attach this chromosome to the spindle when we want to pull the two copies apart. And this chromosome is visible in the light microscope. So you can see a few dividing cells where these condensed chromosomes are visible under the right conditions of staining. Cell division is essential. Remember, all living things are made of cells, right? All cells that we know, if we watch them for a while, trying to discover where they come from, eventually those cells grow, mature, and divide. So every new cell comes from a pre-existing cell. And in fact, it's the DNA in this nucleus that holds the coded proteins that regulates and manages that process of cell division. We start out life as a single fertilized egg, a single cell. And cell division produces 2, 4, 8, 16, and eventually trillions of cells that are exact copies of that original fertilized egg. All of that is involved in growth. In order to study anatomy and physiology, you first have to have a living body with 11 organ systems, all of which have specialized cells that do a certain special job. But each one of those cells comes from the same original cell, the cell that was an identical copy to the fertilized egg. But what has happened in the kidneys is that a certain set of genes are expressed to produce a kidney cell. That process of changing the general cell, the stem cell, into a specialized cell is called differentiation. So this is the way we grow our bodies. Cell division produces the cells. Cell differentiation produces the different tissues, organs, and organ systems that all have to work together to keep us alive. Here's another interesting thing that happens in the cell, and it relates to this DNA double helix that you see over here. This is where a gene, a certain region, holds the code for a specific protein. So how do you go from a gene that's formed from nucleotides? See this sequence, GAG, TAC, GGC? That's a particular gene. How do those nucleotides become a specific strand of protein made of amino acids? This is showing the first step. First thing we have to do is recognize that these two complementary strands are for the stability of the molecule. When they're complementary like this, a C with a G and a G with a C, and it all twisted into a double helix, it's very, very stable in the cell. In fact, when you extract and stabilize DNA, put it in a buffer solution, you can hold it in a refrigerator for years with very little mutation. In order to make a protein, you have to unwrap these two strands. And you'll notice there is one part that begins the coding for the protein, and this other is just the complement. we have a enzyme that manages this process. This is called RNA polymerase because it's going to read this strand of DNA and produce a complementary strand of RNA. Now there's a rub. When we do DNA, A goes with T, T goes with A. G goes with C, C goes with G. But we're going to substitute a new nucleotide, uracil, for thymine in RNA. So the first thing the RNA polymerase does, as it begins to make the RNA, and by the way, if you're asking yourself the question, is this the same RNA that's in an RNA virus? It sure is. The hereditary strand in a virus might be an RNA. But back to this model, here's the RNA polymerase sitting down on a site that says start here. It's called the promoter. You don't need to add an amino acid there. This is the attachment site. And it basically moves to this first triplet. As it moves, it adds a complementary base at each station. So over here you can see the complement is ATG, and by the time this polymerase has moved down to here, you can see that it's added AUGCC. AU, that's the substitute, no T, but a U, GCC. And it's just going to move down this one base at a time, adding the complement. So here comes the next G, and then an A, and then another G, another C, and here's a U again, and it reads all the way down this strand. That produces a strand of mRNA that's complementary to the DNA. The mRNA stands for messenger. The DNA codes for the protein in nucleotide language of the DNA. We're going to transcribe that to nucleotide language in the RNA and move it out of the nucleus in order to translate it to protein. The protein is going to work by assembling the mRNA. This green strand that we just made is the mRNA. It's going to assemble with the blue blob, which is called a ribosome. And the ribosome has these two kind of bubbles drawn in it. These are the binding sites. So if you'll ignore these clover leaves for a second, here's the AUG. And that AUG is a code for a specific tRNA, one of these clover leaves. What tRNA? There's going to be 20. There's going to be a number of tRNAs. What determines which one binds in this site? Complementary base pairing. So if this is AUG, this loop of the tRNA is going to be UAC, and it's always going to carry amino acid number 1. So let's bind this in to get started. By the way, this AUG codon on the mRNA is called the initiator because this is how we start every protein. This amino acid is methionine. But that leaves this second bubble or binding site, and it has CCG. Here comes the GGC tRNA carrying amino acid number 2. If this is GGC, when we're loading this tRNA, we will always put the same amino acid here. So the first step that you can see is to bind both of these sites. So here are two amino acids sitting right next to each other. Once you get that, you're going to break the bond here and shift this amino acid over and bind it to this amino acid. Let's see. And that's what's happened here. Oh, wait a minute. So we're going to shift the amino acid here, and this tRNA is going to be empty. We're then going to slide the ribosome in this direction so that this tRNA, which carries 2 and 1, as a dipeptide is going to enter this site, and we're going to move the ribosome so AGC is now in this second site and ready for binding. So the 3 steps are bind, shift, and slide. Now we're going to keep doing that as long as a new codon comes into this site. So let's go to the next slide now and see what happens after we bind, shift, slide 1 time. We bound this, we shifted that number 1 amino acid over, and we slid the ribosome. So this new codon is in the empty site. Now we're ready for another bind, shift, slide, and here comes the appropriate tRNA. Since this is AGC, this is going to be UCG. By the way, this is called the codon. This is called the anticodon loop. And this anticodon loop is on a tRNA that will always carry the number 3 amino acid. So we're going to bind, then we're going to break this bond between the tRNA and 2 and form a bond, form a bond between 2 and 3. And we're ready to slide again. Now, I've omitted several slides, but the way this process goes is by continuing to bind a codon anticodon with the tRNA and deliver that next amino acid, and then shift the growing chain over to it. And that's going to continue until a particular triplet called the stop codon enters this site. And when that happens, the protein falls off of the tRNA and it's done. The tRNAs, by the way, these empty tRNAs, the cell is grabbing those up and repositioning, putting, for instance, this UAC is going to put a number of methionine right on there, and it's ready to go again. It's reused. So we're going to initiate with this codon, then we're going to bind, shift, slide, bind, shift, slide, bind, shift, slide, bind, shift, slide, all the way down the mRNA, and the stop codon is going to release the protein, the tRNAs, and the ribosomes, all of which can be used again. Now that's a short presentation of that mysterious link between DNA and the MR and the RNA intermediates that deliver the amino acids that make the right protein. It also kind of explains mutation. What happens if an X-ray comes through and breaks this chain? And instead of CCG, this now is CAG. Now instead of amino acid number 2, it's going to be carrying a different amino acid, maybe number 14. Changing that amino acid is a mutation. And that means the protein is going to be shaped differently. It might affect its charge. It might affect its catalytic ability. Might make it so it doesn't work anymore. A lot of pigmentation changes are from inactivating the enzyme. So where it used to make a colored substance, it now no longer makes it, and the pigment comes out white. This is the way a lot of times that you'll get a white flower in a plant that normally has colored flowers. So we're going to continue to emphasize this connection between the hereditary material DNA, its translators, mRNA and tRNA, and the proteins that it produces. Within the cell, things don't just happen. They always happen as a result of that chemistry, that matter, the atoms and molecules, and the forces that are existing at the cellular level. How do things move around? What forces move them? Well, this illustrates a universal force called diffusion. I'm going to define diffusion as the movement of atoms or molecules from an area of high concentration to an area of low concentration. It is a spontaneous force that's occurring all the time. And it is one that, in this model, will achieve a steady state after a while, called equilibrium. Now, this solid, which is a sugar cube with dye in it, In solid state, things don't move much. So that solid sugar cube in a wrapper might sit on the shelf for a long time. But when you put it in a fluid where it dissolves, notice here the high concentration is where the sugar cube is dissolving, and you can see the dye spreading out into the surrounding fluid. Now wait a little longer. Don't do anything to it. And you'll notice that cloud of dye is getting larger and larger as the pile of sugar crystals gets smaller and smaller and eventually disappears. If you wait a long time, you'll notice that the fluid achieves an equilibrium where all of the fluid is pretty well equally covered. This is a result of what we call diffusion. Now, what drives this system? The inherent energy of the system. These liquid molecules have a temperature, they have an energy. So they're going to dissolve this dye and move it at a certain rate. If you raise the temperature of this liquid 30 degrees Celsius, from room temperature of 25 to 55 degrees, then the energy increases, and this is going to dissolve faster and reach steady state faster. Conversely, if you chill this down to 5 degrees centigrade, it's still liquid. It's going to dissolve slower and move slower. Diffusion is going to be effective and more effective as distances get smaller. Diffusion is not going to move me off of this chair. It's much, much too weak a force. But at the level of my cells and the molecules in those cells, diffusion can be very powerful, especially in a system where you're generating a certain molecule at a high rate and you want to transport it out. So if you think about our metabolizing cells, they're producing CO2 all the time. If it gets too high, the CO2 can poison the cell. So the surrounding capillaries and the flowing blood pick it up, and the flowing blood carries it away, meaning the blood that coming into the capillary is low in CO2. High concentration in the tissues from production, low concentration in the blood from being carried away. And so diffusion will always move CO2 into the flowing blood stream. Here is diffusion across a membrane. Now this barrier is going to pick and choose what moves across this phospholipid bilayer. See the phospholipid units with this hydrophobic tail, this hydrophobic zone in the center, and hydrophilic heads that stick out toward the environmental water and face inward toward the cytoplasm. What happens with these molecules? Well, lipid molecules go great through a lipid zone. So cholesterol shown here, it'll just go right through the membrane. However, ions, hydrophilic materials, have to be moved across this hydrophobic zone because water, hydrophilic material, and hydrophobic material repel each other. So we're seeing several examples here. Here we have a protein that forms a channel. Now it's an open channel for the movement of a specific ion. If this ion, you can see the water around it. So the ion has a hydration sphere around it, but it can move through this specific channel. I want to emphasize that it's not just an opening, not just a hole like the hole in a washer. It's actually a specific carrier protein. So when we study nerves and nerve impulses, electrical impulses, we're going to call sodium slow leak channels and potassium slow leak channels that are open all the time and only transport the ions that I named. But in addition, there's going to be a sodium-potassium pump that uses ATP to move ions against the concentration gradient. There's going to be sodium and potassium gates that are different from the first three I've named that will open and close at selected times to initiate the impulse and propagate it along the membrane. And that's just one example. Large molecules that can't diffuse through this hydrophobic zone, the lipids, can't cross unless there is a carrier that allows them access. And so we're going to see a couple of different kinds of carriers. Some need energy, some don't. The relationship of the cell to the surrounding water is a critical relationship because it's not just our cells live in a water solution. That water solution determines, to a great extent in humans, determines the size and shape of the cell. Here's a good example, a red blood cell that has a double concave disc shape. So if you think about a coin, but you take the flat edges and you scoop them, that's what a red blood cell looks like. It's a relatively small cell. It's packed with hemoglobin and it carries our oxygen. Now, in normal blood plasma, the balance between the cytoplasm and the surrounding water medium is at equilibrium. And so this stays a consistent shape. Now, there's a special kind of diffusion here called osmosis. And the movement of water is what is caused by osmosis. Osmosis is the diffusion of water, but across a membrane. What does that mean? That means that water moves in and out of this cell freely, shown by the arrows, if the solution is balanced. And by that, what I want you to do is look at the spacing of the water molecules outside and inside. See how it's kind of equal? So graphically, what this represents is what's called a balanced or isotonic solution. In this solution, this red blood cell will continue to live and carry oxygen and maintain its shape. But what happens if you take a red blood cell? You can do this pretty easily in the lab. Take a drop of blood and start dropping distilled water on it. See how this red blood cell's been dropped in pure water? Notice how on the outside you don't have any solute. By the way, the solute, the purple, outside and inside is balanced in the isotonic. These are the electrolytes and glucose of the surrounding medium, and these are the electrolytes and glucose in the cell. Here we've taken the cell and dropped it in pure water. The concentration, see how the dots move together? The concentration of the water molecules outside are much greater than the concentration inside. So water is driven into the cell. And what does that mean? We don't have cell walls. We just have a cell membrane. And it's like the membrane of a balloon. It has to be supported, but it kind of can flex out. It can expand. And that's what's being shown here. See how it blows this cell up to a larger size, and it rounds it off to a shape like a ball. This is called a hypotonic solution because of the lower concentration of water inside the cell. The cell is hypotonic to the surrounding medium. Now, if you performed this experiment, took a very small drop of blood and dropped on a drop of water, you would see it swell up. Drop on another drop of water and watch an individual red blood cell, and this will swell and swell and swell, and at a certain point, it will burst. So, hypotonic solution bursts your red blood cells. That's just one example of how the concentration of the blood plasma and its balance with electrolytes and other things like glucose and CO2 and oxygen that are all transported in that plasma is critical. The shape of this cell will determine its longevity and also its function. Now let's take this cell again and now drop it in something like a 10% sugar solution. Over here, you'll notice how the water is now farther apart, but there's a lot more solute outside. So the concentration of water inside the cell is hypertonic to the surrounding medium. Since it's higher inside than outside, water leaves the cell, and this cell shrinks. This cell shrinks down to a smaller size, but also you'll notice what happens when that membrane contracts. See how it falls on the cytoskeleton? Those fibers that produce the inner structure, and it produces these little points, these little jagged edges on the red blood cell. Now the red blood cell works because it's basically rounded. These little cells bump along and they're rubbing against each other and they're rubbing against their blood vessels, and they keep moving because they don't stick. Imagine what happens when you have a hypertonic situation and these little jagged, which are sort of like interacting puzzle pieces, like Velcro, grab a hold of each other and block circulation. So hypotonic would burst the cells, hypertonic would shrink the cells. Here's an example of a carrier. This particular protein is specific for glucose. It is embedded in the phospholipid membrane. You can see here the hydrophobic zone is interrupted by this channel. Now I want to first point out, let's imagine this glucose is gone. Imagine it's out here. And when this carrier is empty, it has this shape open to the outside. A glucose enters from the surrounding material and binds here. Now I want to point out something. When you bind two reactants together, you got a new molecule, and that often produces a change in shape. So here is the receptor site, the active site. When it binds, it changes the shape of this, and like a clothespin, it flexes. So the outside entry closes, and the inside opens up. When that happens, the different chemical conditions cause this glucose to pop off. And basically, this is like a rocking chair or a rocking horse delivering glucose back and forth from the outside to the inside, outside to the inside. This is a good example of how diffusion and a carrier can drive this without energy. No energy is needed. When this is released, it pops back open and another glucose can enter and change shape and deliver the glucose inside. No energy is required. The binding and release is energy enough. And it is a source-sink relationship because what? Our nutrients are delivering as glucose all the time. So the concentration outside the cell is going to be high. When you transport glucose in, it's broken down to CO2 and water. It's consumed. So the concentration inside is going to be low, always. And so always this carrier is going to be working to move glucose into the cell. So this is a great example of this term, facilitated because it has a carrier, diffusion because it's moving always from high to low concentration. Just as a sort of a mental note, the system doesn't stop here. In fact, cofactors and assisting enzymes are needed for this to work. And one of the things you need to deliver glucose into the cell is insulin. With insulin present, this transport works. Remove insulin, and it locks this thing in a position that will not let sugar into the cell. That's why diabetics who are insulin dependent, and also type 2 diabetics that are insulin insensitive, cannot get enough insulin get enough glucose into their cells, but in addition, their glucose remains in the surrounding fluids in the blood plasma, so the blood sugar goes sky high. A third example of transport, so we had diffusion, we've had facilitated diffusion. Neither of those require energy. But here's one that is, it's called an active transport mechanism. Anytime you see active transport or you see pump, that means it's going to use a fuel, and the fuel is ATP. The sodium-potassium pump is a big protein that is in multiple copies in every one of our membranes. Every single cell has it. And it produces a state in the body that is body-wide, so that when we look for where is the sodium in the body, It's in the fluid surrounding the cells in high concentration, and it's in low concentration in the cytoplasm. When we ask about potassium, it's the opposite. Potassium's in higher concentration in the cell, in the cytoplasm, and lower concentration outside. So this is called the sodium-potassium exchange pump, and it's complicated. When you use one ATP, so you notice ATP to ADP plus phosphate is not drawn. That's yielding this energy to the sodium-potassium pump. That gives this pump enough energy to move 3 sodiums out of the cell and 2 potassiums in. Burn another ATP, and you move 3 sodiums out and 2 potassiums in. And so the situation when the potassium pump and other carriers are all working together, it sets up a situation with a lot more sodium outside and a lot more potassium inside, but it's not balanced. So there's a charge that exists between the outside of the cell and the inside. That's called the cell potential. We're going to return to that later on. An active transport mechanism, the sodium-potassium pump. How important is it? Right now, if you're listening to this lecture, you're probably at rest and breathing normally. You're relaxed, and all your muscles are feeling pretty good. At rest, a third of the ATP that's being burned in your body is being used only to run this sodium-potassium pump on a constant basis in all your cells. 33% of the energy you're burning at rest is just to run this active transport mechanism that we call the sodium-potassium pump. So transport of individual molecules and ions, we've talked about diffusion, facilitated diffusion, and active transport. But sometimes you don't want to move ions one at a time or molecules one at a time. You're making something and you want to transport them in a mass to another place. A good example, a cell that makes a secretion, something like sweat or milk or oil. We want to transport that to a holding pocket for release in the appropriate time and place. And for that kind of bulk transport, we use vesicles. Now, vesicles are basically just membrane bubbles that are adapted to a specific purpose. And this slide shows receptor-mediated endocytosis to tell you what we know about vesicle formation and transport. Here is an outer membrane. So this is the extracellular fluid, and this is the cytoplasm. And you'll notice these receptors that are mounted facing out. If they're facing out, then the binding molecule, called here a ligand, can bind these receptors. When enough receptors bind, then ATP is expended and fibers form to fold this membrane in. You can see the early stages at 1, the later stages at 2, and then eventually the merging of these membranes to produce a membrane bubble called a vesicle. Now you'll notice when it does that, all that ligand that was bound from the outside is on the inside of this vesicle. So it really represents a capture and packaging technique, this endocytosis, inside cytocell eating. And here's the coated vesicle. Now, many things can happen to coated vesicles. What's happening in this is a lysosome with release enzymes and digestion enzyme fuses with it. And I like this drawing because between here and here, they've retained the portions of the membrane that came from the vesicle, which was at one time this outer membrane, and the membrane that came from the lysosome. You see how that's happened? When you fuse these, it releases the ligand, and they transport it out of the vesicle, the vesicle's now empty, and the ligand might be detached as an independent vesicle, or it may be used for some metabolic purpose here in the cytoplasm. But let's just follow this. Now we've emptied this vesicle out. In this slide, what we've done is detach the lysosome, but we still have this now emptied vesicle with a lot of good membrane and receptors. And following along with membrane properties, the so-called gel mosaic models, we know that lipids can merge and lipids can split, and that's what's happening here. This vesicle merges by touching the membrane and becoming a part of the outer membrane again. It now exposes these receptors to the outside extracellular fluid. And that means they are available for another round of binding and exocytosis. So basically, this can be a cycle of import of this ligand and importing in bulk. You'll notice that the binding is an individual molecule, but we're basically grabbing a whole bunch of them and importing them as a group. This is an example of bulk movement or bulk flow, and vesicles are very important in this role inside of cells. We know from microorganisms, what we call pinocytosis or phagocytosis, that vesicles could be active for that single-celled organism in eating or drinking. Now, we have a digestive system that takes care of our fluid intake and of our nutrient intake. But we also have a need to protect the body, and our white blood cells show these properties of phagocytosis. Here's a bacterium. And the reaction between the surface proteins on the bacterium and this white blood cell cause it to fold in and capture the bacterium, removing the bacterium from free movement around the body. Now a lysosome merges and kills that bacteria and breaks it down. Now there can be some reabsorption of useful things, maybe amino acids, maybe some ATP, but as in all waste production processes, there is a point at which recycling reaches its limits. And this vesicle, now containing the subunits, the parts of the bacterium, but the bacterium is no longer living, it's no longer infectious. And exocytosis, by merging the vesicle with the outer membrane, throws it out of the cell. Another example of bulk movement. DNA again, we can't emphasize it too much. Here's another important prelude to cell division. In order to make the two daughter cells exact copies of the original cell, that means you have to duplicate the DNA, and that's what's shown here. It's kind of like DNA expression, protein production. You unwind the strands, but instead of just transcribing one strand, you copy each old strand. How is it you get an exact copy? The answer is the same: complementary base pairing. So we have here adenine and guanine, orange and green. So here we had, before we unwrapped, this orange was paired with this green. So when we, adenine and thymine, orange and blue, are going to pair, A and T. So this orange was paired with this blue. But when you separate them, making a new DNA segment, you add a blue next to this orange to replace this blue, and you add an orange next to this blue to replace this orange, and all of a sudden what you're ending up with is now two DNA double helixes with the exact same nucleotide base sequence. The old strand acts as a model or a code template for the synthesis of the new strand. And because complementary base pairing is strictly observed, The new strand here is exactly the same as the old strand here. And the identity of the DNA and the genes is preserved. It also explains mutation. Again, what happens if here, where you should have added a green, you make a mistake. Instead of adding a green, you add an orange. There's going to be a kink in this DNA, but in fact, you may complete it. The next time you copy it, instead of being blue-purple next to orange-green, it will be orange-orange, and every DNA that comes from that will now carry a 1 base mutation. The cell cycle is what we observe through the light microscope. We talked about interphase when the cell material is dispersed, and there's a stage of growth, this is the stage of growth G1. This is the stage of DNA replication, where the DNA is copied, and this is a growth stage, growth 2, of protein synthesis preparing for division. Altogether, this is called interphase. The red part shows the condensation of the chromosomes through stages called prophase, metaphase, anaphase, and telophase. Prophase, that dispersed string coils up into the condensed chromosomes. Metaphase, they migrate to the center and line up. Anaphase, the identical copies of the chromosomes separate and go to opposite ends. And telophase, they unwind and form a nuclear membrane to enter the next the next interphase. So we do see these are regulated by a number of factors. Here are chemical factors that affect cell division, and we are going to be studying effects of endocrine hormones on overall cellular effects. But regulating at the cell level is very important to the human body and its maintenance. This growth hormone forms in the pituitary gland at the base of the skull. It stimulates growth, cell division, differentiation in all cells, but especially in epithelium and connective tissue. This is how we get bigger and how we form more skin. Prolactin is from the pituitary gland as well. It stimulates growth, cell division, and development, especially in reproductive glands and in the duct cells and the mammary glands in the female. Prolactin is the hormone that stimulates milk production at the time of reproduction. Nerve growth factor stimulates the growth of neurons and neuroglia, the nervous system. Epidermal growth factor is stimulating to the skin. Fibroblast produces connective tissues. Erythropoietin is a factor that stimulates and increases the production of red blood cells in the bone marrow. We are turning over red blood cells all the time. In fact, millions are turned over every day. So there's always some, but if we move to Colorado Springs, or if we begin to exercise heavily, erythropoietin goes up. So we always have to remember there are chemical factors regulating these cell processes. The final slide is kind of a last lesson. I'll say again and again, why do we look so many times to the chemical level or the cellular level? Why are we studying chemistry in cells to learn more about humans? The answer is, that's where the answers are. Cancer is a disease of the whole body that until very recently in medical history was often lethal. But in fact, it is a modification, a mutation of a single cell, and the effects of producing that one aberrant cell, that abnormal cell, can produce a sequence of effects that can kill an individual. This is the abnormal cell here. The cancer cell starts as a single cell, and in this state, it doesn't really do anything to harm you. But this is a different cell, whereas most of these surrounding cells will divide and then will function for a period of time before they die and required to replace it with a new dividing cell. What happens with a cancer cell is it loses that ability to pause and just begins dividing as fast as it can. So as this disease progresses, the single cell becomes 2, 4, 8, 16, 32, and on up until you get a mass of cells, the primary tumor. Still not very dangerous if it's small, and not very dangerous if it remains compact. So if you see the tumor and you cut out around it and you get every cell, and you basically remove the tumor, and that's a cure. However, things happen where cells begin to invade surrounding tissues. They may invade the solid tissues, connective tissues, and still be relatively local. But because they are displaced from the tumor, surgery may not capture them. If they reach a lymph vessel or a blood vessel and move to other parts of the body, that process called metastasis is the dispersal, and everywhere a new cell lodges, it can produce a new tumor. As long as the tumor is primary and compact, it responds very well to surgery. However, when the tumor begins to disperse, it becomes a whole body disease with multiple tumors appearing all over the body. And this is where an immunotherapy, a chemo, either specific to, if we have an immunogen that can attack a specific tumor cell, or general, where we're trying to kill cancer cells in general faster than we kill normal cells. And these are also cells that respond to and are knocked back by radiation. So all of this results from a mutation that affects the cell cycle. Don't wait for G1, S, and G2. Just go right back into mitosis and divide again. So why do I mention cancer? Because it is a disease that illustrates the importance of the cellular level. It is a loss of a property called contact inhibition. Normally when you form, for example, a kidney of normal size, those cells stop actively dividing. They replace cells that die, but the kidney stops growing. The abnormal cell, the cancer cell, continues to grow a new growth called a tumor. So that completes our review of the cell chapter. And also completes my recording for the pre-recorded lecture.